Hello and welcome to MMI. In this series of tutorial videos, we'd like to show you the basics of the MMI CellCut and how to get good results for further research. The MMI CellCut is our laser microdissection system designed to cut tissues, large regions of interest or single cells, and even living cells for further processing. With its high-pulse UV laser for precise cutting and the patented CapLift technology for reliable tissue and cell isolation, it will help you with all your laser microdissection needs. In this video we show you how to navigate through your samples, set focus points, pins and inspection positions. In Cell Tools there's 4 ways of navigating through your sample. The first way is to click on the hand icon or use your spacebar. Then you can move your sample by clicking and dragging with your mouse any way you want. To get back, just press spacebar or the hand icon again. You can also use the arrow keys on your keyboard to travel short distances on your slide. And you can use the arrow keys on your numpad for larger distances. Velocity and distance of the arrow keys can be defined in the stage movement settings. Here you can define the overall acceleration of your movement, as well as velocity and distance for cursor keys and numeric pad keys as well. If you have a fully motorized microscope, you can also control the Z-focus from within the software. You can either use the slider on the right side, or you can use your mouse wheel. Therefore it needs to be activated. The little mouse wheel on the icon needs to be red. The step size of the mouse wheel can also be configured. Just click on the little drop down. There you can configure the step size, which is very useful when changing objectives. You can also navigate your sample by just double-clicking on the slide viewer preview in the lower right corner. To get an actual overview where to click, just switch to a 4x objective and perform an overview scan by simply clicking on Start Scan. The software now will begin the scan of your whole slide, giving you the perfect overview. Now we can switch back to the objective we want to use. And then navigate the sample by just double-clicking on the structure we want to observe. And by opening the slide viewer, you can also navigate between different slides. This might look a bit different with your setup, because we have installed a long-distance working table here. Here you can also configure your stage inserts by clicking on the little cogwheel icon to access the settings. You can shift them if they do not match your field of view, or you can select different types of holders. Or by directly clicking on the slide, you can select different slide types and shift them in position. Sometimes there might be an interesting structure on the slide that you want to find again later. Therefore, you can pin it by just clicking on the Place Pin button. You can define multiple pins, and via the drop-down on the right, you can get back to them or delete them all. So pin a structure by clicking on the Pin button, move somewhere else, select the pin again, and the software will move back. The inspection position is a special type of pin to inspect the cutouts that you collected with your CapLift. To define it, click on the drop-down, select Set Inspection Position and click on the free space of your slide. Then by clicking on the button, the software will automatically move to that inspection position and move the CapLift down. Click again and the stage will move back to the place where you've started. We hope this video helped you with working with the MMI CellCut. Feel free to visit our other videos for more information about MMI instruments and handling.